Students, let's discuss a very important concept that is the concept of sex determination. That's sex determination in mammals, which also applies to the sex determination in humans. You will see in humans and in all other males, the sex is determined by chromosomes. When the sex is determined by chromosomes, we say this type of mechanism is chromosomal mechanism of sex determination. But before discussing how the sex is determined in humans, we shall first of all discuss as to what is sex. If we, in literal sense, try to define the sex, we say that sex is a set of those morphological, physiological, and ethological features or characteristics which differentiate an organism producing sperms from the organism which produces the ova. It means that sex refers to those characteristics, the set of those characteristics, either morphological, physiological, or ethological, which differentiate an ovum producing organism, that's a female, because ova are produced by females, from the spermatozoa producing animal, that is the male. This is what we call as sex. So, sex is a biological entity having genetic basis which differentiates one individual producing sperms, that's male of the same species, from another individual of the same species producing the female gametes, that's the ova. So this would be the definition of the sex. After that, after completion of the definition of the sex, we shall discuss the mechanism of sex determination in humans. I will tell you that if the sex is determined by the chromosomes, then that type of mechanism of sex determination is known as chromosomal mechanism of sex determination. And in humans and in all other mammals, the sex is determined by chromosomes. So we shall say that in humans, there's a chromosomal mechanism of sex determination. Now the question is, by which type of the chromosomes the sex is determined in case of the humans? I will tell you that in our cells, you will see there are, in human cells, there are 23 pairs of chromosomes. In each cell of ours, there are 23 pairs of chromosomes, and among these 23 pairs of chromosomes are 46 chromosomes. There are 22 pairs of chromosomes. There are 22 pairs of chromosomes which don't bear any relation with determination of the sex. These are those chromosomes which bear the genes for all the somatic characteristics of our body. And these genes which don't have any relation with the sex or which bear the genes which determine the somatic characteristics of the body, they are known as autosomes. So the first category of the chromosomes is the autosomes. Autosomes are those chromosomes which don't have any relation with determination of the sex. They bear genes which determine the body characteristics or the somatic characteristics. And there are 22 pairs of autosomes in case of humans. Now there's only one pair of chromosomes which are related with the sex determination, and this pair of chromosomes is known as sex chromosomal pair. We call them as sex chromosomes. So again I will repeat that there are 22 pairs of chromosomes which don't bear any relation with determining the sex, and those are known as autosomes, and there's only one pair of chromosomes which determines the sex, and that pair of chromosomes is known as sex chromosomes. Now I will try to make you understand that in genetics, this sign, that's like this sign is used to represent the male, and this sign is used to represent the female. So again I will repeat, this is the sign used in genetics for male, and this is the sign used in genetics for representing the female. Now, in case of humans, as there are 23 pairs of chromosomes, there are 22 pairs of autosomes and one pair of sex chromosomes. So this is male and this is female. The 22 pairs of chromosomes, that's the autosomes, will be same in case of the males and in case of the female. So these are 22 pairs means 44A, A stands over here for autosomes. These 44 autosomes would be present in male, and these 44 autosomes will also be present in female. And in case of males and females, they are the same. Remember this point, that in case of males as well as in case of the females, they are the same. So we again say that there are 22 pairs of autosomes which are same in case of males as well as in case of females. The genes which they bear in case of males, they also bear those genes in case of the females, as the somatic characteristics are same in case of males and in case of females. So these chromosomes are same in case of males and in case of females. They bear the same features, they bear the same nucleotide sequence, and they bear the same genes, and they have the same function. These are the genes which don't have any relation with determination of the sex, and they are simply known as somatic chromosomes, or you can also call them as autosomes. This is the majority of the chromosomes in our cells, and there's only one remaining pair of chromosomes which you call as the sex chromosomes. These are the chromosomes by which a male differs from a female. In case of males, you will see there would be the sex pair would be X, Y. So the sex pair of chromosomes in case of males would be XY, whereas in case of females it would be XX. Now, I shall make you understand what does the XX and XY mean in case of the females and in case of the males. You will see the sex pair of chromosomes in case of the females, in case of males is XY. This X and Y has nothing to do with the alphabets of X and Y which you study in English. It means one chromosome is of one type, suppose we represent it by X, and another chromosome is not of this type, which we represent by another letter which we call as Y. Similarly, in case of females, one chromosome is X and another is of the same type, that's also represented as X. So we say that the sex pair of chromosomes in case of the males it is XY, and the sex pair of chromosomes in case of females it is XX. After that, we say that as two different types of two sex chromosomes are found in case of the males, so the sex chromosomes in case of the males are heteromorphic. Heteromorphic simply means there are two sex chromosomes, but they are of two different types. So we say that in case of the males, the sex chromosomes are heteromorphic, that's one is of X type and another is of Y type. Now, in case of females, you will see both the sex chromosomes are of the same type, so we say that the sex chromosomes in case of the females are homomorphic. So with respect to the sex chromosomes, the human males are heteromorphic, and with respect to the sex chromosomes, the human females are homomorphic, or you can also call them as isomorphic. After that, we will see what will happen once the gametes are formed, that is the sperms are formed in case of the males and the ova are formed in case of the female. For example, take the first case, that's of the males. Again, I will say that a male is represented by this sign in genetics. So we shall talk about the males. In case of the males, there are 44 autosomes. So we say the chromosomal formula in case of the males would be 44A. A means autosomes plus one pair of sex chromosomes, that's XY. So this is the chromosomal formula in case of the normal males, normal human males. 44A means 44 autosomes plus XY. Now, once the inside the testicles or inside the testes, the process of spermatogenesis begins, means the gametogenesis begins, the sperms are formed. You will see the meiosis undergoes during the spermatogenesis, and you will see the chromosome number is halved. Now, I will tell you like this. For example, over here, one sperm is formed. We will represent the chromosomal formula of the sperm like this. So, among these 44 autosomes, 22 autosomes would be over here, because in the meiosis the chromosome number is halved, and 22 autosomes is would be there as the chromosome number is halved in case of the meiosis. Now, this process is known as disjunction of the homologous pair of chromosomes, as 22 autosomes land over here and 22 autosomes land over here. Now there is the XY pair of chromosome. It will undergo disjunction. Disjunction means that during spermatogenesis, this X chromosome will get separated from this Y chromosome, and X chromosome will go over here, and the Y chromosome will go over here. So you will see this is 44 autosomes plus XY, 44 autosomes plus 2 sex chromosomes. It is 46 chromosomes, that is 2n diploid number. But as during spermatogenesis meiosis occurs, you will see this sperm contains 22 autosomes plus X chromosome, 1 sex chromosome, that is only 23 chromosomes. It is n means it is haploid. Again, this chromosome, this sperm is also haploid. This sperm is haploid as it contains 22 autosomes plus 1 sex chromosome. This is also haploid. It also contains 22 autosomes plus 1 sex chromosome. But there is difference between these two gametes. Although this is the chromosomal formula of one sperm, this is the chromosomal formula of another sperm, but there is difference. The difference is that this is X carrying sperm and this is Y carrying sperm. So in case of the males, two different types of sperms are formed. In terms of the genetics, one sperm is the Y carrying sperm and another sperm is the X carrying sperm. The X carrying sperm is known as Gynosperm, because whenever this Gynosperm, that's 22 autosomes plus X carrying sperm, fertilizes the ovum, the resultant offspring would be female. That's why it is known as Gynosperm. Whereas this sperm, that's 22 autosomes plus Y, it is known as Androsperm. The andro means male. Whenever Androsperm, that's 22 autosomes plus Y, it fertilizes the ovum, the resultant offspring would be male. That's why it is known as this Androsperm. So now we shall conclude from this point. We shall conclude that whenever meiosis occurs in the testicles of the males to produce the gametes, there are produced two types of gametes in terms of genetics. One X carrying sperm, another Y carrying sperm. X carrying sperm is known as Gynosperm, Y carrying sperm is known as Androsperm. So two different types of gametes in terms of genetics are formed in case of the human males. That is why this process is known as male digamety, or you can also call it as male heterogamety. Why we call it as digamety? The answer is that as two different types of gametes in terms of genetics, that's 22A plus X and another 22A plus Y, are formed in case of human males during spermatogenesis. That is why we call it as male digamety or male heterogamety. Now we shall move towards the process of oogenesis, which occurs in females, and we shall see what is happening during the oogenesis. You will see I will represent a female. This is female. Again, I will tell you that a female is represented by this sign in genetics. You will see in case of the females, there would be 44 autosomes plus one pair of sex chromosomes, that's XX. So human females are isomorphic with respect to, or homomorphic with respect to the sex chromosomes, as both the sex chromosomes are of the same type, that is XX type. After that, what happens in the meiosis? You will see once the meiosis occurs, although in the meiosis you will see from a single cell 4 different cells are formed, but in case of the oogenesis you will see from a single what we call as a cell, 4 different cells are definitely formed, but among these 4 different cells, 3 are polar bodies and only one cell is the ovum. our discussion is not at present oogenesis, but I am giving you a bit information also about the oogenesis, although I am representing only two cells over here. My idea is to make you clear as to what happens in the this what we call as oogenesis, and how the similar types of the gametes are formed in case of the females. You will see once the meiosis occurs during oogenesis, you will see this type of the cell, it will have 22 autosomes plus X, and this will also have 22 autosomes plus X. So both the cells are of the same type with respect to the sex chromosomes. This cell also contains X chromosome, and this cell also contains X chromosome. Among these, only one would be a ovum, that is a separate thing, but you will see only one type of gametes are formed in case of the females. That is, all the gametes which are formed by the female, they will have a chromosomal formula of 22 autosomes plus X. This is what we call as female monogamety. So this is female monogamety, and in case of the males, you will see there is digamety. Two types of spermatozoa are formed in case of males. One 22 autosomes plus X, that's Gynosperm. Another 22 autosomes plus Y, that's Androsperm. But so this is male digamety or male heterogamety. But only single type of the ovum are formed in terms of genetics in the human females. That is 22 autosomes plus this X. That's why we say that males are digametic and females are monogametic, or we say male digamety, female monogamety. Okay, after that, now we shall try to understand if an androsperm fertilizes the ovum, what will happen, and if a gynosperm fertilizes the ovum, what will happen. You will see this is androsperm, that's 22 autosomes plus Y. This is the chromosomal formula of the androsperm. If it fertilizes the ovum, the chromosomal formula of the ovum would be 22 autosomes plus X. Now what will happen? When fertilization occurs, you will see these 22 autosomes, they will form 22 pairs of autosomes or 44 autosomes. These autosomes have, again I will repeat, no relation with determination of the sex. Plus, this is XY. I have already told you that the chromosomal formula of the human male is 44 autosomes plus XY. So we conclude that whenever an androsperm is a sperm which contains Y chromosome, whenever an androsperm fertilizes the ovum, the resultant offspring would be male. It is sometimes also asked in the NEET examinations. So try to remember it very keenly that whenever an androsperm fertilizes the ovum, the resultant offspring would be male. Androsperm is a sperm which contains Y sex chromosome. After that, you will see what will happen if the gynosperm fertilizes the ovum. Gynosperm is the X carrying sperm, 22 autosomes plus X. It is the gynosperm. This is the ovum, 22 autosome plus X. This is the ovum. But I shall make it clear over here that don't get confused that this is 22 autosomes plus X, it is sperm, it's gynosperm, this is also 22 autosomes plus X, this is ovum. This is chromosomal formula. We are only talking about the chromosomal formula of this what we call as sperm and ovum. Rest of the structure is different of the morphological, the physiological structure is different of that of the sperm from that of the ovum. But in terms of the genetic makeup, the gynosperm, you will see it will have 22 autosomes plus X, and ovum will have the same chromosomes, that's 22 autosomes plus X. Now we shall see what will happen if a gynosperm fertilizes the ovum. Again, if a gynosperm fertilizes the ovum, you will see these are 22 autosomes, these are 22 autosomes, these are 44 autosomes plus this is X chromosome, this is X chromosome. So this becomes the chromosomal formula of the female, which I already told you that the chromosomal formula of the human female is 44 autosomes plus XX. So again, try to remember over here, whenever gynosperm is 22 autosomes plus X fertilizes the ovum, that's 22 autosomes plus X, the resultant offspring is always female. So we drew over here two generalizations. That's whenever an androsperm fertilizes the ovum, the resultant offspring is male, and whenever the gynosperm fertilizes the ovum, the resultant offspring is female. Now we will discuss a little bit about the X and Y chromosomes. You will see the X chromosome. This X chromosome was discovered by Hanking. It was discovered by Hanking. And the Y chromosome was discovered by Nittle Stevens. These are the points you should remember for the examinations, as they are sometimes asked. It is just for the information. That's the X chromosomes were X chromosome was discovered by Hanking, whereas the Y chromosome was discovered by Nittle Stevens. Now after that, I will again draw over here the structure. Now we will draw only the structure of the sex chromosomes. In case of the male, you will see male. This is the male. This is the sign for the male. This is the female, and this is the sign used in genetics for the female. In case of male, you will see one chromosome. This it is X chromosome. This is the X chromosome. Another chromosome is this is Y chromosome. This Y chromosome differs from the X chromosome in terms of its genus as well as in terms of its morphology. Okay, so this is X chromosome. This is one type of chromosome. This is Y chromosome. It is different type of chromosome. But you will see there is a homologous region between the X chromosome and Y chromosome that we call as homologous region of the Y chromosome. You will see these are the genes present on the X chromosome and these are the genes present on the Y chromosome. A region of the Y chromosome is homologous with the X chromosome, and we call that region as homologous region of the Y chromosome, as it is homologous with the that region of the Y chromosome is homologous with the X chromosome. So now there's another region of the Y chromosome which contains genes which are particular to the Y chromosome, which are not found on the X chromosome, and that region of the chromosome Y chromosome is known as non-homologous region of the Y chromosome. So this is the homologous region, and this region, and this region, it is the non-homologous region. So again I shall repeat that there are two regions in the Y chromosome. One region is homologous region, which is homologous with the X chromosome, and another is non-homologous region. The non-homologous region contains genes which are particular to the Y chromosome. The homologous region of the Y chromosome undergoes crossing over with the homologous region of the X chromosome during the meiosis, whereas the non-homologous region of the Y chromosome, it maintains the identity of its genes and it doesn't undergo crossing over during the meiosis. After that, you will see the Y chromosome is also known as Androsome, and it is uh, necessary for determining maleness. As it is necessary, as it determines maleness, that's why Y chromosome is also known as Androsome. Now I will draw only Y chromosome, and I will try to make you understand some of the genes which are present on the non-homologous region of the Y chromosome, which are particular only to the Y chromosome. This is Y chromosome, and you will see there are some genes present on the Y chromosome which are only present on this Y chromosome. They are not present on any other chromosome. They are particular to the Y chromosome as they are present on the non-homologous region of the Y chromosome. For example, there is a gene which we call as S or Y. S or Y stands for sex determining region. It stands for sex determining. The sex determining region, it codes for a protein which we call as TDF, that's testes determining factor. That is TDF. TDF stands, again I will repeat, TDF stands for testes determining factor. SRY region means sex determining region of Y chromosome. It codes for a protein which we call as a peptide which we call as testes determining factor. Sometimes this sex determining region is also known as TDF region, is testes determining region. Okay, so it is necessary for determining the development of the testes. I will tell you that until sixth week, of embryonic development, the gonadal ridges are similar, rather same in case of males and females. And after the sixth week is over, in the seventh week, the TDF, that's testes determining factor, it stimulates the development of the gonadal ridge in case of the males into the testes. And due to the absence of the TDF region, upon the X chromosome, the gonadal ridge in case of the females begins its development into the ovaries, although it is a bit late as compared to the development of the testes. So the testes start development in about 7th week, and a week later, or 2 weeks later, the development of the female these gonads, that's the ovaries, starts from the gonadal region. So this is one of the region, this is one of the chromosomes which is, this is one of the genes which is present upon the Y chromosome. Now another gene is known as, which determines porcupine skin. This gene is also present upon the Y chromosome. Now there is another gene, it determines a particular type of the skin which is found in the males, as the skin of the males is different from that of the females. And after that there is another gene which we call as, gene determining hypertrichosis. Hyper means extra, trichosis means hair development of pinna. You will see upon the pinna of the males there are extra hairs, there are more hairs as compared with the pinna of the female. Pinna means the external ear. On the external ear of the males, you will see there are more hairs as compared with the external as compared with the pinna of the females, which have no hairs, and this is rather very less hairs, and this is determined by one of the gene present on the non-homologous region of the Y chromosome, which we call as a gene determining hypertrichosis of the pinna. And you will see these genes which are particular to the Y chromosome, which are present only on the non-homologous region of the Y chromosome. These genes are known as Holandric genes. Whole means whole, andric means male genes. These are it means these are those genes which are present only in case of the males. That's why we call them as Holandric genes. They are not found in case of the females. They are present on the non-homologous region of the Y chromosome. I am again repeating it so that you may remember it. So these are some of the examples of the Holandric genes. That's the SRY region, the porcupine skin region, the hypertrichosis, etc. After that, you will see, now I will discuss a bit about the gonadal ridges and their development. You will see, for example, I draw over here a structure like this in case of females only. When the embryonic development starts, small structures are formed which we call as gonadal ridges. Gonadal ridges. So these are gonadal ridges. This is the gonadal ridge. Sorry, it's not region. It is ridge. Gonadal ridge formed in the male. This embryo. This is the gonadal ridge formed in the female embryo. They are same. Until 6th week, there is nothing like development of the gonadal ridge into the gonads. But once the 6th week is completed, this, in case of males, the Y chromosome, which I already told you, one of the Holandric genes present upon the Y chromosome, that is the SRY region, that's the sex determining region, it determines the development of the male sex. It produces testes determining factor. In the seventh week, the testes determining factor directly impacts the gonadal ridge in case of males, and it stimulates the development of the this gonadal ridge in case of males into the testicles or in the testes. Although it is wholly and solely dependent upon this TDF and SRY region, but one of the epigenetic factors, that is the which is the hormonal factor, that is the androgen, that is the testosterone also help us in the sex differentiation in case of the males. So one is the SRY region produces the testis determining factor, the gonadal ridge in case of the males, it will develop into the testicle or it will develop into the testes. Now in case of the females, there is no Y chromosome, so there is no SRY region, no TDF will be produced, and now the development of this gonadal ridge in case of the females, it will begin to develop into the ovary. But you will see first the development of the gonadal ridge into the in males, first the development of the gonadal ridge into the testicles begins, but in case of females it is a little bit delayed. For example, we say in the 7th week the development of the gonadal ridge into the testes begins, but in case of the females it may be by 8th week or after the 8th week that the gonadal ridge will develop into the ovaries. So some of the geneticists say that female sex is a default sex, although it is a little bit contradictory, but I will discuss over here. Some say that female sex is a default sex. What they mean by saying default sex? It means that this gonadal ridge, it is waiting for the TDF, testis determining factor, which is dependent upon the SRY region. If the SRY region is present, means if the Y chromosome is present, means if the embryo is male type, the TDF will be produced, and the TDF will stimulate the development of the gonadal region to the male sex. And if the TDF is not present, means SRY region is not present, means Y chromosome is not present, which in turn means the this embryo is not male type, it's a female type. Then this what we call as the gonadal ridge will not receive the TDF, and in the absence of the TDF it will develop into the female gonads, that's the ovaries. This is what some geneticists describe as the default sex. They say that by the absence of the TDF, by the absence of Y chromosome, by the absence of the SRY region, the this gonadal ridge in the females develops into the ovaries. This is all about sex determination. But lastly, I will discuss a little bit about the pattern of the sex determination. When is sex determined? With that we will complete. So the question sometimes you are asked in your NEET examination, when the sex is determined in case of the humans. So the sex is determined in case of humans, particularly at the time of fertilization. So the sex is determined in case of all mammals and in case of humans at the time of fertilization. So remember this point. And sometimes it is called as syngametic type of sex determination. So again I will repeat that sex is determined at the time of fertilization in case of all mammals and also in case of humans. This is sometimes known as syngametic type of sex determination. Hope you have understood all about the sex determination pattern in case of the mammals and also in case of the humans.